Hi, my name is Tatiana Casala.
I'm Brennan Harris.
And I'm Leah Hennigan. And for our paper, we decided to look at the relationship between epigenetics and socioeconomic status.
So, previous studies debated the relative contribution of nature versus nurture in human behavior and development, arguing that it was one or the other. But recent research has associated active gene and environment interactions as a key player in human development, and this became known as the study of epigenetics. Epigenetics are phenotypic alterations that occur as a result of changes in gene expression rather than direct alterations to the DNA sequence itself. Therefore, we decided to look at several factors of gene-time environment interactions, such as general cognitive ability, mental health, methylation in early development, and assortative mating, as a function of epigenetic transmission for an individual's future socioeconomic status. And so loosely, socioeconomic status refers to the total degree of economic and social welfare that an individual or family has relative to others in the population. So with that being said, we decided to pose the question, what role does epigenetics play in an individual's future socioeconomic status? And so in order to answer that question, we will discuss the current research and findings.
Of the correlation between mental health and socioeconomic status, the SLC6A4 gene was identified and is known to code for enzymes that break down and promote the reuptake of serotonin. Many studies that have been researching the relationship of child and adult SES focus on the methylation of this gene. One study conducted compared anxiety and depression levels of adolescents in lower SES homes to the methylation patterns of their SLC6A4 gene. They concluded that less methylation of the gene is correlated with negative health effects and a higher risk for mental illness. Ultimately, when the gene is unmethylated, there are too many enzymes working to degrade serotonin, the monoamine that is one of the main deficits seen in those with anxiety and depression. Another study looked at parenting factors along with SES, specifically their protectiveness, and they found that increased protectiveness correlated with better long-term health and higher adult SES for the children. This is potentially due to higher SES also being correlated with increased methylation of these promoters, which prevents too many enzymes working against the serotonin levels. Interestingly, a study found that SES as a child has quite a large impact on methylation patterns compared to the SES as an adult. Meaning if one grew up in a lower SES, but as an adult is more successful, their DNA would more likely exhibit less methylation of the SLC6A4 gene promoters, and this could be due to the amount of promoters that are methylating in childhood is almost double what is active in adulthood.
So in terms of general cognitive ability, in a longitudinal study conducted by Turkheimer et al., researchers collected data regarding socioeconomic status, medical information, and other psychological evaluations on expecting mothers. Once the children in the study turned 7, they were administered the Wechsler Intelligence Scale for children and obtained scores for multiple IQ components. Their findings indicated that 60% of the variance in IQ was due to the shared environment than genes for the low socioeconomic status households, while the opposite was demonstrated in high socioeconomic status households, in which the variance was due to genes rather than environment. These findings suggest that SES modifies the heritability of IQ in children, demonstrating that both genetic and environmental components affect cognitive function. So with that all being said, Leah is going to wrap this up with our conclusions and future directions.
As a conclusion, the studies have shown that epigenetic influences on socioeconomic statuses are more important for children's future than an adult's environment. Specifically, childhood socioeconomic influences adult physical and mental health. As Brennan stated before, it was found that there is a positive correlation between SES and methylation of the SLC6A4 gene, and that methylation of this gene seems to benefit people's mental and physical health across their lifespan. And as Tatiana stated from the GCA study, it suggests that the variance in GCA, such as IQ, is largely due to shared environmental factors in low SES households rather than genes. But IQ in high SES households is more genetic based rather than the environment, since the environment already provides sufficient resources for high GCA. Some limitations we found included assortative mating due to the limitation of interpretation resulting from the mating with similar traits. And adoptive studies also have their limits, such as representativeness, genetic and prenatal environment effects, open adoptions, and selective adoptions. And lastly, the fact that this is a very new field with not a lot of previous research. For the future, we decided that more longitudinal studies can help generate trends or patterns from childhood to adulthood, and they could also provide new knowledge regarding transgenerational epigenetics inheritance. Also, more assortative mating studies can help us as well by identifying the relation of SES to cognitive ability on a more genomic level by looking for similar markers. Overall, it is evident that epigenetic changes in childhood are playing a role in both future health and societal success.
So that is our presentation. Thank you for listening.